the last day that we're going to talk about Punnett squares. I think a week on it is enough to be able to figure out how these things kind of function and work. There's other ways that you can do this, where you can do a heterozygous and a homozygous, as far as looking at something like this, to where the cross would be, if you can figure it out, you would have dominant, recessive, dominant, recessive. Then you would have this one coming across, this one coming down. That would be that, right? And then you would also have another red one to go across to the bottom down here. So if you look at it, you have 50% that would be recessive, 50% that would be heterozygous and showing the dominant one. So you'd have 50-50 for the phenotypic expression. Blue and red, 50-50. But you notice that it's 50% actually heterozygotes, and it's only 50% recessive because you have a recessive parent completely. There's another way that you can do this. No matter what, if you were to do it where you have a homozygous dominant parent, right? And you have a heterozygous one, like this, you're going to notice that this one's going to be homozygous. This one is going to be homozygous dominant. This is going to be homozygous, oh, sorry. This one is also going to be homozygous dominant. And then this one here is going to be what you normally see as far as heterozygotes. You're going to have this one coming down, this one coming across, and at the very end, you have this one coming down, and then the red one coming across to be able to make this. If you look at it, you have 100% that are going to show the dominance. Now, the interesting thing about it is that there are also a couple other things that are happening here. Give me just another moment to express this so we can be done for a while with this. In looking at it, there are things that have multiple alleles. So you might have books that are similar, but they look different. In a certain respect, they have a different allele. You can have rainbows of these different things. All different colors of rainbows of these. You can also have something, and if you're mixing red and blue, codominance, which in this case would be purple. Some of these are just very complicated after a while. So Punnett squares, to me, is a way oversimplification of how this works, especially if you add the epigenetics, which just will blow your mind if you think about it too long.